Madam, do you want me to start?
Yes, sir.
Okay.
Okay, sir.
Yeah.
Okay. Welcome to the day 2, 3 days national e-FDP on recent trends in pharmacological research. Okay, warm welcome to all the participants, guests, all the members in the drug discovery preclinical research phases. In this clinical phase is denoted by the term of the clinical trial. However, only part of the clinical or preclinical research is oriented towards a specific pharmaceutical purpose. The need for fundamental and mechanism-based understanding diagnostic medical devices and non-pharmaceutical therapies means that pharmaceutical research is only a small part of medical research. The increased longevity of humans over the past century can be significantly attributed to advances resulting from medical research. So the benefits of pharmacological research have been discovered by vaccines for measles and polio, insulin treatment for diabetes, medication for high blood pressure. So on like that, so many number of the drugs are discovered, including the major part of the pharmacology is there. So once again, I welcome you all to our national e-FDP program on recent trends in pharmacological research. And I hope we'll have very exciting and enriching sessions in this e-FDP. Now I request C. Abhishek Reddy Garu to speak few words on this occasion.
Thank you, madam. Yeah. Very good morning, everyone. It is my privilege to speak and share my message to all of you regarding today's 3-day national e-FDP on recent trends in pharmacological research. This program focuses on research activities in the field of pharma, which today exigency needs to discover a new molecule for dreadful diseases. To project a novel therapy for any ailment, one needs to go on quest of the therapeutic molecules which cost the basic knowledge on what drugs does to body and body drugs does to drugs in the process of healing. We took the active step of conducting FDP program to cultivate the knowledge to all, from the level of basic to the range of advances in the pharmacological research by introducing the most eminent expertise, commendable professor personalities into teaching all the essential ingredients to the program. I could heartfully hope this program makes you to learn new things for unearthing the innovative molecules in your future studies of research. A continuous promise I hold is ensuring we will conduct coming events like these to deliver and serve knowledge and skill for the happy and healthy society. Thank you. Thank you all once again. Have a great learning experience. And most importantly, in this pandemic situation, I request all of you to stay home and stay safe. And thank you, Rajshekar Rao Garu, for your time and valuable message which you would be giving us. Thank you, sir. Over to you, madam. Yeah.
Thank you so much, sir. Thank you so much, sir. Now I request Chinni Krishnaya to introduce about today's speaker.
It's my immense pleasure to introduce our today's speaker, Professor P. Rajeswara, sir, former professor, University College of Pharmaceutical Sciences, Andhra University, Visakhapatnam, and sir is the president of IPA, AP State Branch. Sir is a well-known person in pharmaceutical field. Sir has received Best State Teacher Award 2004 from Government of Andhra Pradesh, and Sir has guided C.V. Raman Awardee from South Africa, and Sir has funded by the Old Bank entitled Utilization of Weeds of Medicinal Values in Some Important Cropping System of Andhra Pradesh, a NIP ICAR project. The worth of the cost of project is 256.7 lakhs. And Sir is an expert committee member of National Board of Accreditation and former Board of Studies Chairman of Andhra University and Inspector of Pharmacy Council of India. And SAR has guided 17 PhD students and published 53 research papers in reputed international journals and national journals. SAR is a life member of Indian Pharmaceutical Association and life member of Association of Pharmaceutical Teachers of India and life member of Indian Pharmacological Society. SAR is a member of Board of Studies in SRM University, Jawaharlal Nehru Technological University, Kakinada, Sri Padmavati Mahila Vishwavidyalaya, and Chalapati Institute of Pharmaceutical Sciences. And SAR is a member of Pharmacy Council of India Committee for framing course curriculum and norms for infrastructural facilities for B.Pharm.
Thank you very much, Chinna Krishna, the today's convener, for giving the profile of me. Madam Swarnalata, may I start my topic? Chinna Krishnaya,
Sir, please,
may I
sir.
start
So,
my topic?
okay, sir. It's my pleasure to invite for the today's talk, sir. Please start the session, sir.
Good morning, all. Hello. Hello. Is it visible? Hello.
ఎప్పుడు స్టార్ట్ చేద్దాం అనుకుంటున్నారు షేర్ స్క్రీనింగ్. సో మీరు అక్కడ స్టాప్ చేశారు సార్, ఇక్కడ వర్కింగ్ అవుతుంది. మీరు క్లోజ్ చేసండి, ఇక్కడ అంకుల్ దగ్గర నుంచి షేర్ స్క్రీన్
ఇప్పుడు చేయండి. మళ్ళా అది క్లోజ్ చేస్తారే.
ఓకే, ఇప్పుడు స్క్రీన్ షేర్ అవుతుందా?
అవును, నా పీపీటీ రావాలి కదా.
మీ పీపీటీ అక్కడ కనబడుతుంది.
మళ్ళా రీసెర్చ్ కానీ వాళ్ళ చేతనే చేస్తారు. కొంచెం ఇందాక వచ్చింది, వాళ్ళు చేసింది, షేర్ చేసింది.
యాక్చువల్లీ మన స్క్రీన్ షేర్ అవుతుంది ఇలా చెప్పవచ్చు డాడీ, బట్ డాడీకి అంటే వాళ్ళు కనబడరు అన్నమాట. ఇక్కడ వస్తుంది, నీకు కనిపించట్లేదు. నువ్వు ఇందులో చూడు, నీకు లైవ్ లో చూడు, తెలుస్తుంది.
ఏదన్నా రావట్లేదు.
ఇది ఇలాగే వస్తుంది డాడీ, మీరు ఏ విషయం కాదా?
ఏమ్మా వస్తుందా? మీకు అక్కడ స్క్రీన్ మీద కనబడుతుందా? ఇప్పుడు కనబడిందా? ఏంటి? యానిమల్స్ ఎక్స్పెరిమెంటల్ ఫార్మకాలజీ నా ఫోటోతో ఉన్నది కనబడింది. రైట్ రైట్. స్లైడ్ షో ఫుల్ స్క్రీన్ పెట్టుకోవాలి. ఎఫ్ఐ ఎస్, ఫుల్ స్క్రీన్ వచ్చింది. నేను ఇందులో ఏమీ కనబడను కానీ నాకేం. రైట్ రైట్, ఓకే రైట్ రైట్, ఓకే రైట్, థ్యాంక్ యూ థ్యాంక్ యూ. Dear participants, sorry for the delay. Due to a technical problem, a few minutes wasted. Once again, very good morning to you all. Today my topic is animals, experimental pharmacology, its role in drug discovery and development. Research continuously is going on, and the live projects are there, problems that are there, they are answered by the scientists and researched by the scientists, like COVID-19, how our scientists are working. But prior to that, so much of other way of methods are very important in the drug discovery and development. In that particular thing, animals are playing a major role And the knowledge of experimental pharmacology is very much used in the discovery of drugs. And you know, drugs are used for the treatment purpose, and the discovery is a great task. A new drug molecule, if it wants to enter into the market, lot of groundwork will be carried out by group of scientists. Their continuous efforts, innovative thoughts, their hard work, everything resulted in the form of a gift that is nothing but the new drug molecule, and which can be used for the purpose of treatment. In this process, many scientists are involved, like biochemistry, chemistry, pathologists, physiologists, natural chemistry persons, pharmaceutical technology professionals of all forms, pharmaceutical technology, pharmaceutical analysis, pharmaceutical chemistry, pharmacology, pharmacognosy, like biotechnology. Many of the pharmacy professionals are very much involved in the drug discovery and development. Ultimately, answerable molecules are entering into the market, thereby what? Using them, we are alleviating certain diseases and suppressing various pains of the patients. So in this process, preclinical study is very essential, because we cannot use human beings as a subjects in the preclinical study, because we are selfish. But simultaneously, we identified certain animals whose cellular mechanisms more or less similar to the human beings, thereby what? The such type of cellular events and pathological sequences that are happening in humans can be developed in animals, and against that, various lead molecules or the compounds which are thought to be having therapeutic value are assessed first. Why we are doing this particular thing prior to the clinical trials? We are doing this particular practice because the drugs are foreign to the body, and our body is generated from the soil on earth, and it consists all the materials that are present in the nature, and they are taken up, they are used to give over years and years, developed into forms of many lives: animals, humans, flora and fauna. Every cell is having certain genetic material during its evolvement. Through its experience, it developed and generated and made clear to establish its continuity on the earth continuously for propagating and for proliferating its life on the earth. See, the cell can be imaginable with the universe. In the universe, the knowledge what we are having till date is very tiny speck. But when we go on exploring, we are going to know many more things. And similarly, our cell is also having similar position, thereby what? It develops for its survival its own mechanisms where we do not know those things, and any sort of disturbances in that particular mechanism resulted in pathogenesis, and that ultimately causes suffering of the mankind. In order to alleviate, reduce the impact or suppress, certain agents are to be used, and through experience our Ayurvedic gurus, yogis, through their third eye, they found the treasure in the nature, and to the maximum extent during those times, as an Ayurvedic medicine, they prepared and presented to the society for alleviation of suppression or cure of diseases. So in such a way, our natural source is having many answers to the diseases that we are today facing. But we are the poor gentlemen, not skeptically going in detail, and we are unable to find the hidden answers in the nature, thereby what? On and off, we are facing sufferings. For example, in the present COVID, it is a pandemic, and in globally, people are suffering a lot, and we don't know, though they may not be symptomatic in certain individuals, though they may be symptomatic in certain individuals, and they may be serious in several individuals, causing death. We do not know the perfect reasons. But in the past 4 or 5 months, scientists are able to understand, and the basic mechanisms that we understood first is what? Using a mask and using sanitizers and maintaining the hygienic conditions, keeping social distance to the greater extent we can avoid. But if once it comes into the subject, what is the story? And researchers are working day and night, and they are in the process of learning its paths and how best we can avoid. No doubt our internal soldiers, the immunity mechanisms, that may be the cellular, that may be the innate mechanism, whatever it may be. Those mechanisms, when they are activated in us, God has given to us defensive mechanisms. Just like our soldiers in the border, what they are doing against invading enemies. And by using vaccines, what we can potentiate, we can stimulate our internal strengths, thereby what we can fight against that particular enemy. So in this process, even in the COVID, majority of the scientists came to know that in the lungs, in the alveolar 1 and alveolar 2 cells are there. Alveolar 2 cells are responsible for continuously secreting surfactants and protecting the area and helping in the transfer of oxygen from lung tissue to the blood component, thereby what? Oxygen saturation to the extent of 97 to 99% achieved, thereby oxygen supply to the whole body is present. And he is performing because oxygen is very much required for the metabolic function. Continuous oxidation is going on in that, oxygen is very much required in order to make alive all the, and also in order to stimulate the functionaries properly, thereby what? Toxicant molecules will be relieved from the body and the internal environment cleaning can be made. When the internal environment cleaning is made, naturally the cell very healthily it works. Very healthily it works. So in such conditions, what? We must do a continuous effort in researching to get lead molecules which are showing minimal toxicity and maximum efficacy. You know, these molecules are toxic in nature because they are foreign to the body doesn't want, and if at all in a negative energy way, if those molecules activate certain systems, a negative functioning will be there that promotes the pathological condition and appearance of a particular disease. For example, Aurora A kinase. It is very essential in mitosis and proliferation of cells. But when overexpression is there, then cancer appears in many forms. Breast cancer, lung cancer, cervical cancer, gastrointestinal cancer, whatnot, in many forms, bone cancer, like that. So, if we know those pathways, if we go into the Viswam, what we call universe, and if we understand in its fluid the reactions that are happening and the pathways, altered pathways that are there, naturally they are the targets for the drug molecules. So in such a way, drug discovery and development continuously, it is an effort for the good of the mankind. But in that, animals are the candidates, helping candidates who are involved in every drug molecule till date we are having. Without animal experimentation, there is no drug at all. FDA will not accept even. And this is the prior this thing, before entering into the subject, let us overview our cellular structure and its function. You please
Cells
follow the
are
video.
The smallest living units of an organism. All cells have three things in common, no matter what type of cell they are. All cells have a cell membrane, which separates the inside of the cell from its environment. Cytoplasm, which is a jelly-like fluid, and DNA, which is the cell's genetic material. There are two broad categories of cells: The first category is eukaryotic cells. They have organelles, which include the nucleus and other special parts. Eukaryotic cells are more advanced complex cells, such as those found in plants and animals. The second category is prokaryotic cells. They don't have a nucleus or membrane-enclosed organelles. They do have genetic material, but it's not contained within a nucleus. Prokaryotic cells are always one-celled or unicellular organisms, such as bacteria. So, what are organelles? Organelle means little organ. Organelles are the specialized parts of a cell that have unique jobs to perform. Let's start with the nucleus, the control center of the cell. The nucleus contains DNA, or genetic material. DNA dictates what the cell is going to do and how it's going to do it. Chromatin is the tangled, spread-out form of DNA found inside the nuclear membrane. When a cell is ready to divide, DNA condenses into structures known as chromosomes. The nucleus also contains a nucleolus, which is a structure where ribosomes are made. After ribosomes leave the nucleus, they will have the important job of synthesizing or making proteins. Outside the nucleus, the ribosomes and the rest of the organelles float around in cytoplasm, which is the jelly-like substance. Ribosomes may wander freely within the cytoplasm or attach to the endoplasmic reticulum, sometimes abbreviated as ER. There are two types of ER. Rough ER has ribosomes attached to it, and smooth ER doesn't have ribosomes attached to it. The endoplasmic reticulum is a membrane-enclosed passageway for transporting materials, such as the proteins synthesized by ribosomes. Proteins and other materials emerge from the endoplasmic reticulum in small vesicles, where the Golgi apparatus, sometimes called the Golgi body, receives them. As proteins move through the Golgi body, they're customized into forms that the cell can use. The Golgi body does this by folding the proteins into usable shapes, or adding other materials onto them, such as lipids or carbohydrates. Vacuoles are sac-like structures that store different materials. Here, in this plant cell, the central vacuole stores water. Going back to the animal cell, you will see an organelle called a lysosome. Lysosomes are the garbage collectors that take in damaged or worn-out cell parts. They are filled with enzymes that break down this cellular debris. The mitochondrion is an organelle that is the powerhouse for both animal and plant cells. During a process called cellular respiration, the mitochondria make ATP molecules that provide the energy for all of the cell's activities. Cells that need more energy have more mitochondria. Meanwhile, the cell maintains its shape through a cytoskeleton. The cytoskeleton includes the thread-like microfilaments, which are made of protein, and microtubules, which are thin, hollow tubes. Some organisms, such as plants, that are photoautotrophic, meaning they capture sunlight for energy, have cells with an organelle called a chloroplast. The chloroplast is where photosynthesis happens. It's green because it has a green pigment called chlorophyll. Plant cells also have a cell wall outside of their cell membranes that shape, support, and protect the plant cell. Animal cells never have a cell wall. There are many other unique structures that only some cells have. Here are just a few. In humans, for example, the respiratory tract is lined with cells that have cilia. These are microscopic hair-like projections that can move in waves. This feature helps trap inhaled particles in the air and expels them when you cough. Another unique feature in some cells is flagella. Some bacteria have flagella. A flagellum is like a little tail that can help a cell move or propel itself. The only human cell that has a flagellum is a sperm cell. In summary, remember, eukaryotic cells are plant and animal cells with a nucleus and membrane-enclosed organelles, while prokaryotic cells are unicellular organisms without these things. All cells have a cell membrane, cytoplasm, and genetic material. And even though only plant cells have chloroplasts, both plant and animal cells have mitochondria.
Well, now you are very clearly knowing about the cell nature and its function and how it cleans the garbage, daily garbage, and how it maintains the homeostatic conditions, and thereby what? The metabolic activities are constant and life of the cell is maintained well. But when a foreign particle, which is foreign to it, when it enters, it damages that particular environment by many ways, physicochemical means, thereby what? Disturbing the several pathways in the cell. They themselves then responsible for the development of certain diseases. So in the universe, how we do not know completely about it. Similarly, in the cell also we do not know completely about it, but we know some pathways, some receptors, and some kinase enzymes which are involved in the metabolic process and all. Here, the topic is recent trends in pharmacology, recent trends in pharmacological research. And my subject is basically, without understanding properly the insight for the future developments, naturally that may bring wrong energies, thereby what? Majority of the finding may be not to the true extent. So in this way, my way of discussion will go on animals, experimental pharmacology, drug discovery and its role in drug discovery and development. But the webinar topic is recent trends in pharmacological research. And just we outlinely we touch the recent trends in pharmacology and drug development. Majority of the teachers may be there, or some good viewers are also may be there. And nowadays, what we have to do, we have to select a live project of the recent trends. Then we contribute something to our pharmacology as well as to the support the community as well as. So we have to take such a problems for solving them In that binding kinetics and pathways of ligands to GPCRs, G protein coupled receptors, that is one open channel for the discovery of drugs. As I mentioned, the Aurora A kinase, a priority pharmaceutical target for treatment of cancer. This pathway is very important because its overexpression leads to produce and develop several forms of cancer. So this is one trend to be touched out in order to make smiling of cancer patients. The another trend is also there, that is what? Novel adjuvants designed for improving vaccine efficacy. And it is very important. And in these lines also, some scientists, these adjuvants, they are using and they are potentiating the efficacy of vaccine. They are immunomodulators, immunostimulants, the potentiators, immunopotentiators. And in that, several forms are there. You are all teachers, you may be knowing. A good pharmacologist, I think. You may be knowing the salts of calcium, alum, iron, zirconium, and certain polysaccharides. They are all used in this particular efficacy improvement research, these adjuvants, and research work is going on in this particular way in order to improve the vaccine efficacy. So this is one trend and one line waiting for the discovery of drugs. The next is target protease activated receptors. PAR1, 2, 3 are there, and they are all activation leads to inflammatory condition, and this is one target trend awaiting to the interested researchers to tap this particular line. And other thing is what? Risks associated with new drug development. See, drugs earlier I mentioned, drugs are foreign to the body. And just now you have seen the cell. Our body is made up of the things that are there in the earth. And mostly organic and inorganic, both are there in our body. And electrostatic force is there. Charge is there. And each molecule, a different charge will be there. So that what? We are all, our total body is a charged body. By way of simple polarization and depolarization, it is doing many things. And you know, even release of neurotransmitter, certain signal is to be there. And finally, at the end, the depolarization is responsible. The inbuilt mechanisms activated and releases the neurotransmitter from the vesicles that are stored in stock and release it nearby and interacting with many receptors, centrally as well as peripherally. And similarly, but their charge and skeleton very much suitable to interact with only one type of protein component. For example, adrenaline is there. It is having its affinity to interact with alpha, beta, beta 1, beta 2, beta 3 and alpha 1, alpha 2. And a small structural changes from adrenaline to isoprenaline, making a charge difference, and simultaneously adrenaline on all adrenergic receptors have and isoprenaline only on beta receptors. So in such a way, they are changing. When charge is changing, when the bulkiness of the molecule changes, when the side chain alters, so much of pharmacological nature of the molecule is altering. For example, morphine is a narcotic analgesic. But at the amine level, if we made it into the tertiary amine into quaternary amine, immediately the pharmacological nature of the molecule is altered. It is a narcotic analgesic. When it is quaternized, that nitrogen, it becomes a neuromuscular blocking agent. See how morphine is converted by simple mechanism of structural alteration and charge modification into a separate category of skeletal muscle relaxant. So similarly, these drugs, when we are developing, sometimes they are good, sometimes they are bad. But when we are developing a new drug molecule, it is also when it is going to the universe, that is in our body, it is having many places to shake his hands. And sometimes the shaking hand will be a good wishes, and sometimes the shaking hand damages. So ultimately, the person will be sufferer during the treatment itself. Sometimes they may be very serious, sometimes they may be moderate, and sometimes they are mild. And mild to moderate type of adverse reactions can be, risks can be tolerable, tolerated. But whereas severe risks, very difficult to tolerate, and here a vacuum is there where one can find a good molecules or antidotes for the adverse reactions. In the clinical pharmacology also, lot of vacuum is there where that is also a good area for the research. Recent advances in psycho and neuropharmacology is also another good area. And another thing, recent toxicological studies and its issues, that is also another new trend where one can tap. So those are the trends like that. Even in COVID, I told you alveolar 2 cells where the angiotensin 2 receptors are present, it interacts. And here we have to find out certain agents where they can stop this particular interaction, thereby what? The damage that is caused through the alveolar 2 cell, type 2 cells can be minimized, thereby survival rate of pandemic may be increased. And here, through re-engineering mechanisms, some scientists, they found that this vasoactive intestinal peptide and its analog may be useful at this particular juncture, and research is going on. Thereby what? That is also one particular area, a new trend in pharmacology for the discovery of drugs. And you know, we are all pharmacists. And what is the role of pharmacy professionals? See, my intention is what? In the earlier slides, I have given the way of doing research at the higher level, and student level, the role of pharmacy professional is also very, very important. I hope undergraduates, graduates, and teacher friends, research scholars may be participated in this webinar. And combining all these things, what? I am giving a generalized talk not focusing on a one particular discipline to make you to enlighten. And here, first we have to know the role of pharmacy professionals. The role of pharmacy professionals, the quality of medicines supplied to patients, ensuring that the supply of medicines is within law, ensuring that the medicines prescribed to the patients are suitable, advising the patients about medicines, including how to take them, the reactions that may occur, and answering the questions which were asked by the patient. It is a very important duty of a pharmacy professional. Advise other healthcare professionals about the safe and effective medicines use. Provide service to the patient. Example, the pharmacy professional can do smoking cessation, cholesterol management, blood pressure measurements, and this sort of things and their impact on health. If we educate the community, urban as well as the village, naturally that will be a good way of serving as a pharmacy professional. And he is having another important duty, these pharmaceutical technology people. They have to supervise production, preparation of medicines, and also the quality of medicines. And here, he is a guarantor to the public that the drugs that are prepared are genuine, they are quality medicines, thereby what? They can be used without any fear effectively. Thereby, in this process, this pharmaceutical technology person, what? He assess quality assurance starting from the production to the end product release. Simultaneously, other professionals also involved in quality control at each and every step. Ultimately, a quality product we are going to dispense. And here, the other things that he has to see related to the marketing wise, friability and dissolution rate, disintegration rate, and not only that, these things, and dissolution studies are also to be carried out. And you know, majority of the products that in generally that are prescribed by the doctors are oral drugs. So when we are taking oral, it enters into the gastrointestinal tract, stomach, and it disintegrates, dissolution, then in the ileum, at various sites it absorbs, and this process is going on. And here we must know about the physicochemical nature of the compound, and to safeguard the in acidic media, how it is to be uncoated, the technology is to be used, and site delivery drugs also can be developed. In such a form, all these things will be developed. And prior to that, what happens when it is going into the stomach, I want to show you through a video. You please follow it.
After swallowing an aspirin tablet, it will go on a journey through your digestive system. Absorption. The tablet will travel down the esophagus into the stomach, where it is dissolved into much smaller particles. These particles will continue their journey into the small intestine, where most of them will be absorbed into the blood. The small intestine walls, also called lumen, are lined with tiny villi which stick out to provide a large surface area for the nutrients inside the food to be absorbed. Looking closer at these villi, you can see they have a rich blood supply and they are covered in a thin layer of epithelial cells. The aspirin molecules will work their way through the epithelial cells and into the bloodstream, where they are carried around the body. Distribution. Blood plasma contains complex molecules called albumin. Albumin is a protein made by the liver which transports nutrients around the body. Albumin also binds to drugs that enter the bloodstream. Here you can see some of the aspirin molecules binding to the albumin. All the aspirin particles that have been bound will be filtered out as they pass through the liver, and only the remaining aspirin molecules will get through and have any effect on the body. Different drugs will bind to albumin in different amounts depending on their affinity for plasma protein. The next stop on the journey is the liver. The aspirin-rich blood will enter the liver through the hepatic portal vein at the bottom, and it will exit at the top via the hepatic vein. At this stage, even more of the aspirin will be removed through a process called first-pass metabolism. The liver's job is a bit like a filter. It will metabolize drugs before they reach the rest of the body. It does this in a few steps. Phase 1 metabolism. First, aspirin is hydrolyzed, breaking off ethanoic acid and leaving behind salicylic acid. This salicylic acid isn't water-soluble, so it can't be excreted in urine yet. Phase 2 metabolism. An ionized group is then added to the salicylic acid, forming glucuronide, which is water-soluble and can be excreted in urine. A lot less of the aspirin will continue its journey onto the rest of the body, including the site of the pain. Only 68% of orally taken aspirin will have any effect at all, because of all the filtering that the body has done. This percentage is called the drug's oral bioavailability. If drugs are not administered orally, e.g. intravenously or buccally, then they can bypass the small intestine and liver, so none of this filtering takes place, and they have 100% bioavailability. We are now at the site of some skin tissue damage. Tissues in the skin are creating prostaglandins, which are causing inflammation. The prostaglandins are sending messages to the nerves in the skin responsible for the sensation of pain. Once the aspirin arrives in the blood supply at the site, it prevents the production of prostaglandins in the tissue, which will reduce the swelling and stop the messages being sent to the nerves that are perceived as pain. Excretion. Any remaining aspirin in the blood will go round and round the blood supply, going back through the liver several times until it is all used up or filtered out of the body. It travels into the kidneys and then into the bladder for excretion.
So, keeping in view these points, the pharmaceutical technology people, they develop formulations and they enhance the bioavailability of the drugs by many means. Hence, a competition is there, bioequivalency and bioavailability improvement by the traders, saying that particular point they are improving their business. So that is how that technique works out. Why therapy is required? Why drugs are to be discovered? We know various pathological states are there, various disease forms are there to us. We are encountered with them. Sometimes we are battling positively, many times negatively. So, to improve battling power of the patient, drug treatment is very essential. So thereby, specific drugs are to be discovered. When we are discovering drug, definitely adverse reaction also will be there. They are also to be taken into consideration, and drug discovery program is to be carried out in such a way to reduce the adverse drug reactions and to improve the efficacy of drug activity. So what is disease? Till now we are talking about disease. What is disease? A disease, an abnormal condition affecting the body of an organism and associated with the specific symptoms and signs. For example, if prostaglandin levels in the parietal cells affected through that combination is there, H2 prostaglandin receptor in the wall of the acid secreting cells. And here, when acid production is more, naturally, when bicarbonate production is less, there is every chance for burning, gas formation, and finally ulcers formation, and pain in the stomach or bleeding of blood in the tools. When tools come out, they may be appeared as a dark brown in color. That is an indication that internal bleeding is there somewhere in the gastrointestinal tract. So this sort of things will be there. So a disease is an abnormal condition affecting the body of an science. And experimental pharmacology is what? It is a method development through which certain experiments can be made in order to screen the biological activity of the drug molecule, whether it cures, repairs, or slows down a particular thing. So in our laboratory, this particular technique we have developed, and here the heart is beating at 360 level, 300 to 360 level rate minutes beats per minute, and in that particular condition, thoracotomy and tracheotomy is carried out, and oxygenation is given to the animal, and the pleura is taken out. Then the descending coronary artery is found. At area risk, a knot is developed with the help of a thread, and it is double knot method we develop, and we can stop at where we want to stop the blood circulating to a particular part of the heart there. Then after half an hour of blood stoppage, then we started reopening the canal. Then reperfusion will be there, and that time we have given nearly 4 hours. And the aim of the experiment is what? To find out the infarction that developed due to the reperfusion injury. During the reperfusion, free radicals are generated, and the experimental protocol in that what? Are there any drugs prior to the heart operation? If they are treated with them, naturally the blunting of operation procedures can be minimized, thereby benefit to the patient can be made. With that thought, we developed this particular 2-knot coronary artery ligation method, and we were successful. And the research that is published in Artery Bypass, it is also texted in the Artery Bypass textbook in the chapter 14. That is a American, the publishers there, and that credential we got by doing research on this particular method. And this is elephantiasis, say disease. So many types of diseases are there, and for all, drugs are needed in order to alleviate the pain of the patient. Even diabetes, diabetes due to the insulin reduced sensitivity, or it may be due to the overexpression of insulin and burning of the islets of Langerhans, insulin release block, or whatever it may be. During that time, properly the glucose is not entering into the cell. As you see that biological cell membrane is this is lipoidal in nature, bimolecular lipoidal in nature, and it works as a barrier to from one cell to the other, and fluids that are there, interstitial fluids are there. And drugs, when it is circulating through the blood, it crosses that and reaches inside when it is desired into the cell. And if it is stopped, naturally the drug response will be decreased. And similarly, if glucose transport is stopped, though the glucose transporters 1 to 5 are there, they may be working out, but they may not be working out with full kinetic form. Thereby what? The blood glucose levels increases. When blood glucose level increases, naturally it glycosylates several proteins and different forms of disease conditions it develops. And especially in the case of the eye, what? Diabetic retinopathy. This is the normal vision. This is the retinopathic vision. Thereby what? That is a defect developed due to a disease. So treatment is required. And similarly, diabetic foot ulcers, immunity is greatly decreased, and a small wound may become a big wound, and it may be sometimes the leg is to be taken out from the individual due to the sepsis. And gouty arthritis, like many disease forms are existing. For all, we need treatment, and for the treatment purpose, we need drugs. And even kidneys also damaged by the diabetes. And high sugar level also may cause hypertrophic cardiac myopathy, where the heart pumping greatly reduces, the ejection fraction also greatly reduces. If you see the normal with the hypertrophied heart, you see the thickness of the walls, even auricle, greatly thickened, and the ventricle size is decreased and volume is less, and effective force also developed in that ventricle is less, thereby ejection fraction greatly decreases. It should be more than EF, ejection fraction, more than 70, or even 70 is good. So now, the pathological development of various disease forms we touch. And here, why I am showing all these things is what? First, we must understand the disease form, and for which, how a solution to be developed. And if we know the pathological condition, pathological sequences, and the metabolic alterations that are responsible for a disease condition, then we can identify the targets. Thereby what? Targeted based drug discovery can be made. For example, in the stomach itself, hyperacidity you take, and acid secretion is regulated by several mechanisms. Gastrin is there, calcium is there, acetylcholine M3 type of receptor involvement is there. For the enterochromaffin cells, release of histamine need the cholinergic stimulation, and there the M1 receptors are present, and that histamine acts on H2 receptors on parietal cells, and thereby that is a G protein coupled receptor, thereby lot of acid production will be there. And to counter this particular imbalance, the God has created another system besides that, and that is called prostaglandins. And prostaglandins are continuously synthesized there, and it is having its own prostaglandin receptor. When prostaglandin synthesis, when acid secretion increases, its synthesis increases, and simultaneously that is also a G protein coupled receptor. It inhibits the activation of H2 receptor, adenyl cyclase. When that is inhibited, naturally histamine stimulation inhibited, this adenyl cyclase responsible for the development of second messenger called cyclic AMP. That cyclic AMP in the cellular universe, it interacts with a kinase system. That kinase phosphorylation resulted to activate proton hydrogen pump, ultimately acid. When it is inhibited, acid production is final common pathway. That is one target. That is why omeprazole, esomeprazole, like many aprazoles are there. They are the targeted, drug targeted synthesis will be carried out. Thereby, new drugs can be developed with minimal side effects. And say, that is one example, and ischemic heart diseases. Say, hyperlipidemia is one factor, smoking is another factor, and food habits are also another factor. Like many factors are there, which may be damage the endothelial lining and rupture and clot formation. Finally, occlusion of the artery takes place. See, this is the left descending coronary artery. I told you earlier in my experiment, ischemic heart disease experiment, we found out this left descending coronary artery. We ligated at this particular position. This is the area at risk. We calculated like that. And some of our compounds during my research, they showed prominent, and I told you it is also national, international reputation it got, that work. And here, when this particular area is damaged and it occludes there and blood supply is stopped and ischemia develops in that particular area, and proper survival mechanisms if not taken place, then slowly the infarct area develops and finally it becomes big, so that that area will be there in the person but as a non-functional unit. And ultimately that infarcted area, what it is doing, it is actually reducing the ejection fraction. That means what? Circulation rate is decreased, and finally due to this heart stoppage also maybe will be there, that ultimate death it results. So these factors, disease development, here a thrombolytics treatment is to be given in order to, if the clot is more and cannot be using the thrombolytics, repair cannot be made, then bypass grafting is to be conducted in order to survive the patient. So these are the maneuvers, medical as well as the treatment through drugs. Blood thinners, using blood thinners, we can improve. And here, earlier I told you about the peptic ulcers, and now you see the disease, how acid is formed in our stomach.
Hydrochloric acid is produced in parietal cells through a complex series of reactions. Carbon dioxide diffuses into the parietal cell, and the enzyme carbonic anhydrase catalyzes a reaction between the carbon dioxide and water to form carbonic acid. Carbonic acid dissociates into bicarbonate ion and hydrogen ion, and the bicarbonate ion is transported back into the bloodstream. An ion exchange molecule in the plasma membrane exchanges bicarbonate going out for chloride coming in. The hydrogen ions are actively transported into the duct of the gastric gland, and the negatively charged chloride ions diffuse with the positively charged hydrogen ions. Potassium ions are counter-transported into the parietal cell in exchange for hydrogen ions. The net result is production of hydrochloric acid in the parietal cells and its secretion into the duct of the gastric gland.
So this is the models, experimental pharmacology, the development of ulcers in the stomach, and afterwards the drug which is having anti-gastric ulcer activity can be tested. And in our laboratory study, we conducted with certain drugs and found very good. And yeah, so the earlier thing is what related to the disease and the trends in pharmacology research and the professional activities of a pharmacist we discussed. And the science in generally, first it is developed by the forefathers of pharmacology, the pioneers of pharmacology. Claude Bernard, a French physiologist, he identified, he is from 1817 to 1883, survived. During his time, what he identified the mechanism of curare, the arrow poison. He found that the animals are paralyzed due to the blockade of neuromuscular junction when curare arrow is arrowed to it. And he is one who developed the idea of pharmacology and the mechanism that is there with a treatment, the concept, basic concepts and all, and first person to develop interest in this particular part of science. The next one is Rudolph Bukim. He is a German pharmacologist. He is also to that particular age, 1820 and 1879. He lived up to 1879. And he is the person for the development of bioassays. And his students, Oswald Schmittberg, who masked his guru's this thing name, and he contributed a lot in pharmacology. And he is responsible for isolation of certain neurotransmitters, epinephrine from pituitary gland, histamine isolation, pure crystal insulin isolation, like many works he carried out, which are the mark bench in the field of pharmacology. Next person, John Jacob Abell, the father of American pharmacology. He also, person, contributed a lot towards the pharmacology. The other person is from India, Sri Ramnath Chopra, who worked mostly on herbal drugs. And he is the person for the development of Central Drug Research Laboratory in Lucknow. Earlier, he worked in Calcutta as a professor of pharmacology in Calcutta University. Simultaneously, the Central Laboratory at Calcutta also he controlled. And to his greatness, he worked a lot on Rauwolfia serpentina and on herbal drugs. And he is the father of nation, India. And in his commemoration, the Postal and Telegraph Department released a stamp on Sri Ramnath Chopra for his contributions. They are the pioneers of pharmacology, given insight in pharmacology to us and developed. And now, in the initial stage, I told you that preclinical study is very essential. Without preclinical study, no drug molecule will come out. And similarly, in the earlier days, by using the animal models only, many drugs are surfaced. For example, in 1796, vaccine for smallpox developed. The sacrificer is who? Cow. In 1881, vaccine for anthrax developed in sheep. In 1885, vaccine for rabies developed in dogs and rabbits. The subject is what? Animal. In 1905, pathogenesis of tuberculosis developed in cow and sheep. Then the targets are determined and drug development taken place. In 1999, mechanisms of immunity elucidated in guinea pig and rabbit. See, they are the subjects for the development of that. Insulin discovered in dog in 1921. 1928, pathogenesis of typhus discovered, guinea pig, rat, mouse. 1932, functions of neurons discovered in cat and dog. 1942, the RH factor discovered, rhesus monkey. That is why RH factor is given to that. It is discovered using rhesus monkey, thereby what? RH factor that is given. In 1943, vitamin K discovered, rat, dog, mouse. In 1945, penicillin tested in mouse. In 1954, polio vaccine developed in mouse and monkey. Open heart surgery developed in dogs. It is carried out in open heart surgery. First, it is developed in dogs. And then subsequently, after acquiring the technology and the technique skill, then slowly it extended. Now, open heart surgeries are very frequently, it is carried out in many hospitals for the survival of patients. And in 1964, regulation of cholesterol discovered in rat. In 1968, rubella vaccine developed in monkey. In 1970, lithium approved in rat, guinea pig. In 1975, interaction between tumor virus and genetic material discovered, monkey, mouse. In 1984, monoclonal antibodies developed. The animal is mouse. In 1919, organic transplantation technique advanced using dog. In 1995, genetic transfer of cystic fibrosis developed, mouse, non-human primates. Monkeys are used. 1997, prions discovered in characterized hamsters and mouses. In 1998, nitric oxide as a signaling molecule in cardiovascular system discovered in rabbit. You know that nitric acid is a vasodilator, and majority of the pharmacologists, the regions and the mechanisms for relaxation of the vascular smooth muscle is known, and this particular pathway mechanism discovered using rabbit. So that means what? Majority cases what? These developments, animals are being used and they are sacrificed. Their sacrifice and the results we are refined, and now we are using for our survival, all those things. And in 2000, brain signal transduction discovered in mouse and rat. 2002, mechanism of cell death discovered in worm. 2003, non-invasive imaging method, MRI, for medical diagnosis developed using rat. You see, animals are helped us. In 2004, odorant receptors and organization of the olfactory system discovered using mouse. Say, in all the discoveries, animals are the first subjects through which we are getting certain results, and those results, after refining and improving the quality and all, we are using for the purpose of human elements. Now, why drug discovery? It's necessity. For our country, drug discovery is a compulsion. Majority of our available drugs are from foreign pharma companies. See, the drug discovery at hot rate, it is not going in India. If at all, a one or two molecules that made by Sun Pharma, none other than that, no molecule is developed in India, but re-engineered molecules are very well developed in India for the purpose of business. So, the reasons are also there because a lot of expenditure incurred in the development of drug discovery. Majority of our available drugs are from foreign pharma companies. This has not only created our dependence on foreign pharma companies, but also produced adverse effects on our economy. Because we have to pay for them and we are using their drugs. That means what? Whatever the profit the business people here they are getting, but simultaneously the same type of profit is given to the foreign companies. That means what? We are affected economically due to this particular problem. So, drug discovery is very essential in order to improve the GDP or economy of our country. Our scientists should geared up to meet the challenges. So, animals, experiments and obligation. Why we have to do prior to the clinical trials, animal experimentation. Animal experiments. One hand, we are emphasizing the need of drug discovery, and on other, animal experimentation is being opposed. This is a glaring contradictory situation. To discover and develop a drug, experiments on efficacy and toxicity are very essential. See, I told you the drug molecules which are going to be discovered, they are foreign to the body and they are not exactly interacting at the site at lowest concentration, because the bioavailability concentration to reach the site to the maximum level in order to suppress or cure the disease, we need large amount of drug. If at the site the same amount of the drug is available but improved technology and techniques, naturally paracetamol need not be taken 650 milligrams thrice a day. In the hypothalamus region where the thermoregulatory center is there, where the prostaglandins playing a role in this particular heat dissipation mechanism, if technology developed, a few micrograms or sometimes a nanograms may be sufficient. So, and toxicity of the drug also will reduce. To discover and develop a drug, experiments on efficacy and toxicity are essential. Let us know about the essentials of animal experiments for drug discovery. Drug discovery and development. In that, first, identify the disease. It may be ischemic heart disease, or it may be a prostate cancer, or it may be a breast cancer, or it may be bone cancer, or it may be a ulcer, or it may be a diabetic condition, whatever it may be that. First, in all these cases, the disease is to be identified. Once the disease is identified, the biochemistry, pathology related with the disease, and the cellular mechanisms that are altered by certain proteins, they are to be identified. And when they are identified, then it will become very easy for us to target a molecule. So in the drug discovery, identification of disease is very essential. And here, the isolation of protein involved in the disease, and it takes much time. As I told you, the protein, it may be a protein kinase, or it may be a MAP kinase, or it may be a Aurora kinase A. Whatever it may be, the protein that is there, that is to be discovered first, and then other combinatorial or in silico or by direct synthetic process, whatever it may be, through many mechanisms, the targeted molecules can be synthesized. Thereby what? Drug molecules can be made. So now when the isolated protein involved in the disease, when isolation of protein and the mechanisms, if we know, then find a drug effective against the disease protein. It takes some, it is a time consuming time, one, a 2 to 5 years it will take. Then next, then when we feel that yes, this molecule definitely targets that area, then we must understand. And why animals are being, because their cellular mechanisms and our cellular mechanisms to the maximum extent they are similar, and that is why we are taking animals, and preclinical testing is carried out. It takes 1 to 3 years. When we start a project, we will, when this particular place comes, now time adjustment and everything we will do in such a way in order to reduce the final end product to be delivered, and preclinical study is carried out. After preclinical study, if proper information comes, yes, it is definitely working as an anti-ulcer activity or anti-diabetic activity, targeting the or sensitizing the receptors, insulin receptors, or improving the glucotransporters activity, whatever it may be. So in when that is there, naturally the drug in scales that will be scale up, and formulations are also will be developed. When all these things are developed, the total information that is there filed, and investigational new drug, as an investigational new drug, it works out and it is submitted to the FDA. And after that, going through it, and human clinical trials approval will be given, and human clinical trials will be carried out. It may be 2 years or it may be many more years, the human clinical trial, depending on the urgency and need of the drug molecule. See, for example, in the COVID, if I want to develop and clinical trials are to be carried out for a period of 2 to 10 years, it is waste. And in such conditions, FDA also regulates and they give time slot for that particular type of things, because here the first priority is what, if it is showing any good, naturally on the same subjects a treatment can be started, and thereby what, if it survives, that is a positive side to us, and also for the development it helps. When that is there, after information, phase 1, phase 2, phase 3, clinical trials, after the molecule, the simultaneously, when we are doing preclinical testing, simultaneously toxicological testing also will be carried out. And toxicology reports, all things will be filed, and along with the IND that will be submitted to the FDA, and FDA also verifies all those things, and human clinical trials it gives access, and then after human clinical trials again file NDA, new drug application, that it has provided uh, uh, good control and the molecule is safe to use for a particular disease, and it will be submitted to the FDA, and FDA will go through, and then it will give permission and approval will be there, and this approval will take 2 to 3 years depending on the necessity and clarifications, and it takes 2 to 3 years. Then it will enter into the market, and that means what? Starting from first year to nearly a 15 years time it will take, and sometimes high throughputs when we are using, it may be shortened in 10 to 12 years that project may be completed, or 13 years the project may be completed. When 13 years if a project is completed, naturally the patent approval for a years, so that the original company which spends a lot of amount on the development of this particular drug can recover, and based on that only the rate of the drug also will be there. And drug design, molecular modeling, visual screening, that is all at that particular find a drug effective against disease. And technology is impacting this process. You have seen that nearly a 15 years time it has taken, or even more also, and when if it is 15 years, 5 years time will be there to the producer, and if it is 16 years, 4 years time will be there. How he will recover that? Afterwards, after 20 years, the many pharma companies will be focusing on the molecule, and prior to that only they will be re-engineering those molecules, and they will also file, and then it will be the property of all, not the property of the original bidder. So in that what technology is impacting this particular process, lengthy time can be shortened. Identify disease, isolation process, finding drug, and preclinical testing. So here the genomics, proteomics, bioform potentially producing many more targets and pharmacological targets. Thereby what? We are getting a clear cut picture here, and how I have to attack that particular problem in order to alleviate the disease, in order to suppress the disease, or in order to cure the disease. And high throughput screening also can be carried out through that information also supports. Thereby what? The time gap that is there, the time that is taken in the previous session of discussion, and now can be shortened using technology. So the technology is impacting this particular process. Thereby what? In the shorter duration we can complete the project. Thereby what? Longer time visual screening. Longer time can be allotted for the marketing purpose and all. Thereby the individual mother company will be getting to some extent profits. So technology is impacting this particular process. Now majority of the drug discovery programs are going using high throughput screening only, and thereby the time slot is also reduced. This is how in the discovery of drugs, different groups will be involved, and they are chemists, biochemists, pathologists, pharmacologists, and microbiologists, whatnot. Many engineers also, certainly engineering people also will be involved. So first, when we want to work on a particular disease, all these groups will be sitting, programs will be made, and finally each group will entertain that particular problems related the work, and thereby what? Togetherness, finally it will come to a position in a short time when all are working, all groups are working. Thereby they can reduce the time frame required for the development of a new drug molecule. So technology is giving a great impact on the development of drugs. So overview of drug development. Stage 1. This is our overview till we discussed, and in the stage 1 is what? Hit and lead compounds development. It may be from natural source. Till date, nearly 65 to 70% of drug molecules are from the natural source only. Innumerable number of compounds are derived through the natural source only, and everything is done at this particular stage 1. In the stage 2, preclinical studies, when the set molecules are developed, immediately stage 2 preclinical studies will be carried out, and then phase 3 clinical studies are also will be carried out. So this phase 2, phase 3 clinical studies called experimental pharmacology. The information that given a dose in the clinical study, the minimum dose that is required, the repeated doses that are to be given over a period, this sort of information, and the compatibility of the drug with the subject, and any variability is there, any toxic effects are seen during the course of treatment, even in the phase 3 clinical trials, because in the disease condition, how fast actually it is during the disease, is there any variability from person to person, if at all the variability in time frame, if that is there, what is the reason, is the genetic component or the other factors that are like this. Total information will be studied by the clinical pharmacologist in their study. So this is related overview and experimental pharmacology. And now, importance of preclinical study in drug development. And yes, why we have to do preclinical study in drug development? So it is very important, it is needed. And in the preclinical study in drug development, it involves mainly experiments with animals and it is useful in characterization of toxicity profile. That is very important. The thalidomide case, especially it is given to get sleep, and mostly pregnant women will have sleep disturbances, and several gynecologists, they prescribe this particular drug, thalidomide, and after some time it appeared that those persons who taken the thalidomide got the teratogenic babies and limbless, that is the organ dysfunction, and this sort of things are happened, and uh, the company actually stood before the court and lost its all property for the simple reason. So toxic profile is very important. If it passes the toxicity profile, then only it will be allowed to work further, and thereby what? Toxicity is very important, so that by animal experimentation, characterization of toxic profile, that information will get, and safety and efficacy of drugs, that information also will get, and dose determination also will get, because in acute toxicity we will get LD50, based on the LD50 1/10 or 100 taking doses, and further work will be carried out, and thereby what? We get a idea related to the dose that is to be given, and when it comes to the phase 1 clinical trials, during that time further correction can be made, thereby what? Whether which dose at what concentration it is to be given, and that sort of information, dose determination, that is the another important thing. By conducting preclinical study, we get information. Then what is an animal model? Why we have to use animal? If it is happened in the animal, will it happen in us? How guarantee? How we can say extrapolate those results to human beings? This sort of questions may arise. But according to Wessler, 1976, a living organism with an inherited, naturally acquired, or induced pathological process that in one way or another closely resembles the same phenomena in humans. So that is that type of similarities are there. Thereby what? If we do, definitely to a greater extent we will get answer to the new drug molecule, so that whether it is to be carried further work or not can be decided and judgment can be made. Thereby what? Animals are being used in the preclinical study because of this particular phenomenon. Drug discovery importance of animal testing. India's rich natural resource, it is a great boon to us, but we are not to the greater extent working on this particular thing. Still, we are lagging in identifying the molecules. And from the natural product chemistry will work, synthetic chemistry will work, combinatorial chemistry will work, so that the prospective molecules will be identified, and they may be working for a particular disease pattern, like that it will be settled. And when this is there, next phase is what? Other studies will be implemented. Structural function and activity. That is the physiology and biochemistry will be studied. And for that what? Animal testing is needed. And pharmacodynamics, physiology, pharmacology, immunology. What happens? Animal testing is required in the as a preclinical matter. Pharmacokinetics properties, whether how the pharmacology as well as the physiology and enzymology and all these aspects will be studied. And in vivo activity, physiology, behavior, pathology, pharmacology, in vivo activity studies that gives an idea related with the activity of the molecule. And safety, this is more important. Physiology, metabolism, how it metabolizes, whether the metabolic products are having any toxicity uh, or safety. These aspects are also will be studied, so that for this what? Animal experimentation is very essential. So we have to use animals in the drug discovery program. And the 3 R's in animal research. We are taking the 3 R's in animal research is what? An important criterion which all scientists engaged in animal research must follow, are 3 R's. 3 R's, the first is refinement. Then the reduction in the model number, in that particular model number, reduction to the maximum extent. And if at all any death is there, replacement. All things are contributing a lot in the animal research. And thereby what? The criteria is what? Refinement, reduction, and replacement. 3 R. 3 R's are very much important while we are doing drug discovery research on animals. What animals we use and why we use them? This is the another. What animals? And I suppose the viewers and listeners may be there. I don't know to what extent they select the animals for a particular model of disease, and how far the results that you are producing using animals acceptable at higher bench, higher level. So all these things are to be focused, and one should know about the use of animals. It is very important, very, very important. And here, in that what, more than 90% of animals used for drug research are rats and mice, as they represent mammalian group. They are available through laboratory breeding facility. Only 1 to 2% drug research is conducted with large animals, that includes dogs, cats, and primates. The scientific reasons behind using them are our cardiovascular system is closely similar to dogs. Our nervous system of cats resembles to that of the human beings. 95% of our genes are similar to apes. So finally, a drug molecule, when it is set, and finally it will be studied even in monkeys, and only the information will be given, because and 95% of genes of monkeys and ours are same. So on these reasons what we use animal study, because our cardiovascular system is closely similar to drugs. Our nervous system is closely related to cats, and 95% of genes are similar to apes. These are the things. So that is why we use them. Now we are focusing our attention on what type of animals we have to choose, what type of experiments can be carried over those animals. How far our research work we are doing is standard using the animals. You can assess after seeing this. See, experimental animals are vertebrates and invertebrates are there. The vertebrates: rodents, mouse, rat, guinea pig, hamster, gerbil, and non-rodents: rabbits, cats, dogs, monkey, etc. Others are frogs, zebrafish, chicken. They are also can be used. So these class of animals are being used in the experimental pharmacology. Approximately 10 million vertebrates are experimented around the world every year, and this is the percentage follow-up of those particular groups in the animal experiments. And now rat, you see regularly, you are seeing in your laboratory, this is albino strain, and subgroups are Wistar and Sprague-Dalley rats. And here Wistar rats, they are having wide head and the ear is long, whereas tail length is less than the body length. By observing the animal itself, we can say whether it is a Wistar rat or a Sprague-Dalley rat. When the tail length is less than the body length, naturally that is a Wistar rat. And wide head will be there, ears are long to them. And the rat that is appearing here in the picture is Wistar rat. Sprague-Dalley rat, longer and narrow head, tail is longer than the body length. So that is Sprague-Dalley rat. And one particular point you must keep all the time in mind: what? They do not have tonsils and gallbladders. Please don't conduct any experiment related with them, gallbladder or tonsil. And they are having diffuse pancreas. So not a good model for type 1 diabetes. Type 2 diabetes experimentations can be made. But type 1 diabetes, for that what you should not use. Tail of rat helps in thermoregulation. So this is the rat you have seen. This is one animal used in the experimental pharmacology. Then inbred strain. So now you are seeing dark agouti rat. They are inbred rats, and research application of this particular inbred rat is urinary bladder tumors. This is the best model for study urinary bladder tumors. And further, this model can be used autoimmune diseases conditions. This model can be used in multiple sclerosis, collagen-induced arthritis. So dark agouti rat, these rats are very costly. Even mice, if we buy a specific strain, it may be costing you 1200 to 1800 per mouse. And another inbred rat is spontaneously hypertensive rat. This is a Sprague Dally rat. See, the length of the tail is more than the body, you can see. And the research application is model for insulin resistance and essential hypertension in non-obese humans. So such type of research, you have to use spontaneously hypertensive rats for insulin resistance studies and non-obese humans. Now you can assess the research that you have conducted. If you do in higher centers in industry, no doubt definitely they follow all these qualities and specifications. Thereby what? International community will accept the results that they are showing. Thereby what? Further promotion will be there. And in your research, now you think whether a proper animal that you have used for your research or not. Childhood hyperkinesia and ADH conditions also, the spontaneously hypertensive rats are used. Screening antihypertensive drugs, this particular, when the drug availability, for example, if it is from a natural source, availability of the drug is very minimal, a few mg are there. And by way of local as well as the Ayurvedic information is available very highly on that as an antihypertensive, so that those even low concentration that is there, in such cases this spontaneously hypertensive rats can be used for screening antihypertensive activity. Otherwise, dog and cat are very good models. Now this is brown Norway rat. This particular rat. This is also inbred rat. The research applications of this particular inbred rat is behavior, myelocytic leukemia, mercury chloride induced nephritis, and in cancer research, these particular type of rats are to be used. Brown Norway rat. See, variability is there among the inbred rats, and selectivity is also varied. So variation is there in selection. So specific animal is to be taken for a specific disease condition. Thereby what? The we are shown to the international community or to the higher link up desk. Thereby what? Authentically, with strong evidence, we can say that yes, our studies shows that this molecule is definitely having this particular activity, and this molecule is very competitive and a candidate for IND. And now outbred strains. Outbred, again research applications, this Vistar and Sprague Dally rats we are using regularly in our laboratories. And Vistar is for teratogenicity study, carcinogenicity study, liver physiology studies, evaluation of anti-ulcer drugs, isolated rat preparations for identifying many things, even autopoids or new chemicals that are released from a particular preparation, and where there is no proper analytical and testing that is not there. So isolated rat preparations like uterus, colon, and ileum useful for bias of various neurotransmitters. So Vistar strain is used. And Sprague Dally neuroendocrinology studies, memory studies. See, when anyone is doing on these memory studies, it is better Sprague Dally rats use. Don't use Vistar rats. Ischemia study, you use Sprague Dally. Neurological effects of nutrition, induced excitotoxicity, nociception and signaling. In such research, Sprague-Dally rats do good. This is long Evans rat. This rat application, research application, is preferentially alcoholic embryopathy in leukemia and breast cancer studies, diet-induced obesity studies, neurological research, diabetic research, behavioral research. In such conditions, what? This long Evans rats are being used. That is its research application. And next, mutant type of rats are also used in research. Zuckerberg rat. This is, see, now you have seen many types of rats now in research application. And this Zuckerberg rat, important genetic model for research on obesity and hypertension. So in such areas where they are very much interested on hypertension and obesity, the Zuckerberg rat is the only model for doing research. It may cost even 5,000 each rat. And another is what? Outright nude ethnic rat. The research application of this nude ethnic rat is most preferred model for immunological research. If you are doing any immunological research, this nude ethnic rat model is the authentic rat model. And cancer research also. Tumor angiogenesis studies, hereditary kidney diseases. In all these applications, these nude ethnic rats are being used at the higher level. And mouse. So this is a very widely used animal in our laboratories, smallest laboratory animal, and Swiss albino mice, nude mice, hairless, genetic mutant, bridge mice, lack natural killer cells and or susceptible to cancer. So different mice are there. Smallest laboratory animal, common strain is that Swiss albino mice, nude mice, big mice, and mice used for their tail to help in thermoregulation. Research applications of these mice, a variety of classes that we have seen, they are mostly used in acute toxicity studies, screening of various pharmacological agents like analgesics, epileptics, etc. And teratogenetic studies are carried out in mice because easily handling and so they are being used. Next animal is guinea pig. Yes, guinea pig is another model. This is also, we are in generally in regular laboratories also, college level laboratories also, use it for the experimental purpose. This is the best model for screening bronchodilators, immunological research, study of tuberculosis, screening of spasmodic, antispasmodic compounds using isolated preparations. The intestine in generally used isolation preparation for many purposes and for many biological assays. This is one type of animal being used in experimental pharmacology. And this is hamster, and two types are there: golden hamster, Chinese hamster. Experimental tumor mainly used in diabetic control research, where this Chinese hamster mainly used in diabetic research, used in cytogenetic research, good model for Duchenne's muscular dystrophy, DMD, and golden hamster is being used in experimental tumor production, hormonal effects on reproduction, diabetic caries, cardiac myopathy, polycystic diseases. And those in dental research, naturally, this particular golden hamster being used for dental caries. Jerpil. Its application is good model for stroke, used in lipid metabolism and heart disease studies, obesity and insulin resistance, diabetic cataract and angiopathy, vertebral degenerative studies also, this particular animal being used. And rabbits, you people know very well, and it is there in every laboratory, very handy and very friendly animal. So many types of studies can be carried out using this particular rabbit. Even screening of mydriotic meiotics can be made, and local anesthetics can be screened. That intestine can be used for identification of many chemical components of the body fluids, and even enzyme induction inhibition studies can be carried out. And see metabolic studies when we are doing in intraurethral cannulation technique using intra, that is developed in our laboratory. Continuously urine outflow will be there. Through urethra we will insert a cannula, and finally we lock that so that contraction is obstructed. Thereby what urine samples can be collected continuously, and the metabolic studies can be carried out at what rate, and kinetics can be using urine data can be studied, and rabbits can be used. So applications are many, and it is a very good model in laboratory practices. And cat, it is I told you, it is on CVS pharmacology. Cat is the best animal. Apart from that, in many screening models also it can be used. For example, ganglionic blocking agent screening, and neuropharmacology, oncology, chromosomal abnormality studies. In many studies, what cats also used as a model for screening. Dogs, this is also best model for cardiovascular. It is very good model. The cervical sympathetic and vagal nerves are run together. Hence, stimulation of nictitating membrane through preganglion sympathetic is complicated by central vagal stimulation, causing reflex variation in blood pressure. That will be there. So that cat is the best model in that particular thing for the ganglionic blocking studies, but dog is not to that extent because of this variability. For it is also used for many forms of experiments can be carried using this particular dog model. And finally, I told you animal experimentations will be carried out using many types of drugs. And finally, these monkeys, because 95% of the genes are similar to the human, so they will be subjected on mice, and then the information will be provided to the FDA, thereby this investigational new drug application can be made for the approval purpose. And frogs are also used, for many purposes, and as a learning purpose to the students, that there are non-cholinergic areas are also there where acetylcholine can be synthesized using frog, we can show. And all parts can be used, even for drugs as cardiotonic drugs also this can be used. And what are most commonly used in CVS and CNS study. For retinal toxicity of drugs also, frogs are used. Isolation organs like rectus abdominis muscle and other parts use it for screening, skeletal muscle relaxants, and for finding the agonistic components that are there, compounds if it are there, mimic or relaxing gastrointestinal tract. Screening can be made using them. And zebrafish. Eggs develop outside mother body and different developmental stages can be observed under microscope. Useful in studying the effect of drugs and development of embryology and genetics. So zebrafish in such things what zebrafish is a model and it is very essential even in research. This is also to be carried out. Any genetic abnormalities are there and developmental embryology phase what happens. Use it for toxicity studies. This is also used for toxicity study. In air pollution studies, all these zebrafish is very much useful. Even we can use this as a toxicity study because the volume of drug will be minimal. And pigs are another one. And pigeons. Here, pigeons preferred in screening antiemetic activity. Cardiovascular diseases such as spontaneous arteriosclerosis and standardization of cardiac glycosides, pigeons are used. CNS such as anxiety pigeon method, apomorphine-induced stereotypic behavior in pigeons, screening of IV anesthetics, bioassay of prolactin. For this sort of things, pigeons are being used. Now it is very clear to you that in the drug discovery, animals are very important, and we must know what type of animals are to be used for a particular purpose, whether we are using the quality animals for getting the standard results or not. And you people have seen many animals are there in the drug discovery program, and for a specific program, specific animals are being used in such a way. Preclinical studies are carried out. Now, benefits available due to animal experimentation, that is, a medicine, Accutane and Proaccutane, has been used to treat skin diseases. During animal testing, it was found to cause birth defects, and for this reason, doctors never prescribed this drug to pregnant women. Many cytotoxic drugs developed for dealing with the cancer are found to be highly toxic in animal testing system. So, the benefits that we are toxicity, if at all there, that we are finding out, and thereby what proper precautionary measures and any alternative chemical alteration will reduce that and improve the efficacy of a drug molecule. This sort of things will come out, and thereby what this animal experimentation gives a lot of information, and through which we are benefited in altering and getting a true form of molecules in the drug discovery. And 30 to 40% of drugs are found to be unsuitable at their advanced stage of development due to potential toxicity and high risk for human life. This clearly elucidates the importance of animal testing. So the importance here clearly tells us that, and doing animal experimentation to the maximum extent, drugs are screened and we are going to identify whether they are toxic to the humans to the extent to damage and to promote to the level of death or not. There, if at all that sort of toxicity arises with that molecule, immediately we can discard and we can save the time working on the same molecule for further duration. So in such a way, the importance of animal testing, it clearly illustrates that the toxicity and the high risk for human life can be detected. Thereby what precautions can be taken. Failure of drug due to insufficient animal research. That is also another thing. If we do not do proper investigation using animals, we may be losing a good molecule. Sometimes a toxic molecule may enter into the market, and after a period it may show its toxicity, thereby what we have to discard that drug molecule. And ultimately, if it is within 1 year or 2 years, naturally the time, money, everything is wasted. So it is animal research program is very important in drug discovery. The failure of drugs due to insufficient animal research. Here, the most important example is the thiazolidine class of drugs, rosiglitazone. Use of this drug has been found to be associated with risk of cardiovascular events like congestive heart failure and myocardial infarction. Another TZD and pioglitazone has very recently reported to induce bladder cancer and osteoporosis in long-term treatment. That is why they are to be discarded. The therapeutic potential may be there, and simultaneously the toxicity that appears in the subjects is a hallmark for discarding that particular drug. European Medicine Agency on 23 September 2010 recommended the suspension of the marketing authorization for the rosiglitazone anti-diabetic medicines. India has finally banned the much controversial drug rosiglitazone prescribed for type 2 diabetes. Later, we lost our valuable natural resource production due to lack of research drive for drug discovery. See, not only we need continuous drug discovery and research, it should be a continuous program. If we do not do, naturally we are losing a valuable natural resource. Not only that, loss of money also will be there. Curcumin from haldi is an excellent anti-cancer compound with which human trials have progressing at NIH. Curcumin is now a property of America. See, we are using that, and that is the compound of us, but due to lack of our research, we are unable to patent it, and curcumin is now a property of America. Similarly, we will lose many others if drug discovery in our country is not given a serious attention along with the animal testing facility. Our dependence for drug will not only cause economic loss, but also the irony of fate is also valuable natural resource materials of potential drug candidates will be the property of other countries, and we will buy them paying a high price, rendering them unaffordable for several suffering common people of our country. So, drug discovery must a program in India, thereby what? Our resources can be come to limelight, thereby what? The progress of GDP as well as the benefit to the diseased persons will take place, and thereby government should think on it. So now you have seen many animal models which are being used for different purposes, and now handling the animal is also very important. See, we should not roughly use the animals, and the psychology and psychological attention of the animal during the experimentation should not be disturbed, and you should not feel tension, and all factors will be there, so that handling is a technique through which we can make friendship with the animal, and we can do our experiment. Animal will be silent, and our experiment will be conducted very easily. Handling and restraining of experimental animal is an important aspect in practical pharmacology, induces feeding treatment or any procedure with animal. In India, CPCSA has given complete guidelines for animal caring, housing, and handling requirements. This is also very important to know how to handle the animal. So, mouse handling technique. See, with the thumb and ring finger, what we have to hold the neck of the animal and without giving much tension on the animal, and we have to handle, and intraperitoneal injection can be given or oral feeding can be made. Rat handling technique. This is how with these two fingers and thumb finger below and the four paws are there, we have to hold the neck and four paws in such a way not to give much movement and not to give much strain on the apply much strain on the animal. This is how we have to handle the rat. And guinea pigs handling is to be carried out in this way. Rabbit handling techniques. Say rabbit, we have to hold the rabbit in the shown way and not to create any tension or discomfort and to give discomfort to the animal. This is how you have to hold the rabbit. Route of drug administration. There are different routes are there and you all learn. Amma, iదేంటి షాక్ వస్తుంది ఏంటిది తీసే అక్కడ ఏమైనా తగిలిందేమో షాక్ వస్తుంది ఆఫీస్ తీసే Intraperitoneal injection to different animals. See, this is how we have to give intraperitoneal injection. You see, the way the needle tip is actually introduced into the animal. And if it is at the center point, if you give the position here, there the bladder will be there. Instead of intraperitoneally, the drug may go into the bladder. So, intraperitoneal injection in different animals holding, this is mouse, and in rats also holding properly we have to give. And when we draw the plunger, air will come. That is an indication that we are giving only through intraperitoneally. If it is in the bladder, naturally urine will come out. Intravenous injection through tail, ear vein. This is how, that is to the mouse or rat, tail vein, intravenous injection, marginal ear vein injection. This is how we have to give. And intramuscular injection, this is how we have to hold the animal, and to the lower limb thigh we have to give injection. Gluteus muscle in the rabbit we have to give injection by holding the animal in that particular position. And subcutaneous injection, nape of the neck, there in the mouse we have to give, and nape of the neck of rat and flank of the rat, this subcutaneous injection can be given. And this is how the intracardiac injection in rat is given. And the first second from the lower, first second and third rib level, if we pierce the needle inside, it will go to the ventricle. Intracardiac injection can be given. And this is the experiments I shown you that on rat, cardiac surgery rat, I have shown you earlier. So in my lab, using the minimal subjects, I carried out research. So some of the, I say, the instruments that are being used in drug discovery laboratories or even industrial research laboratories are at the high end, and some of the isolated heart experiments are carried out, and this tracheal chain, exposure of the tracheal chain experiment, pharmacology, learning how to expose the trachea. This is how in rat models we do. And in our laboratory, the 150 mg of heliotropium indicum given a very good anti-ulcer activity when compared to the earlier I shown you the inflammation of the total stomach was observed, and now at 150 mg kg, dose-dependent effect is seen, and see 150 mg, the stomach layers are very clear and there is no ulcer at all. And some of the equipments used in industrial this thing where I visited and where they are doing an acute asthma studies, and they have used many things. Acute asthma studies models, and this is how the intranasal injection that is to be given, how I can drop and automatic organ bath. They are so these are some of the industrial equipment in generally used for the experimental studies. And you people know, sometimes you may not be knowing the nature of compound activity, and you are blind, but people may be claiming that it is having certain activity. In certain cases, what the screening methods are there, screening. And screening is what? In three forms. One is simple screening, blind screening, and programmed screening. So in the drug discovery, simple screening, when a particular area is there where the plant products are having certain anti-diabetic activities thing, but there the area is very big and we have to identify the plants which are having anti-diabetic activity. In such cases, what? We offer only a simple screening, only blood reduction levels only will take it into consideration whether it is having any anti-diabetic activity or not, whether it is reducing the blood glucose level or not. Though it may be having many other important activities, we will not mind for them. Only this particular screening only we will do for rapid identification of potential plants. So that particular screening is called simple screening. When simple screening is over, in the programmed screening, when it shows a good results, a 30 to 40% reduction in blood sugar levels, then a programmed screening will be carried out. In the programmed screening, again, in diabetic models, it will be carried out. And there is a sequence of series are there. A battery of tests are to be carried out in program screening, and in that, using that particular methodology, the program screening will be carried out, and definitely it gives whether the compound is possessing real anti-diabetic activity or not. So that gives a promotion as a candidate to participate in drug discovery laboratory information. When there is no information, a blind screening will be carried out. In the blind screening, here the scientist as well as the local people may not be having any information regarding its medicinal activity, but some clients may be there. So here what he will do, in order to, many potential activities may be there. Here he will, using simple techniques only, he has to identify the scientist, and here he do a blind screening. And blind screening, what, based on, without using any sophisticated or high throughput screening methods, he will identify the pharmacological categories that are shown by the molecule or the plant extract or the isolated compound from a plant. And here a scoring will be there and profiles will be there. The profile is what? This behavioral profile, neurological profile, and autonomic profile. By doing the blind screening, there we will get the LD50 values. And in that way, afterwards, a programmed screening will be carried out. And finally, whether it is suitable for the drug to enter into the drug discovery program or not can be assessed. So this is how the drug discovery program is going on. This is how the animals are being used in drug discovery program. Many variety of animals are there. They are used for a particular purpose, and that type of work is to be carried out using that type of animals only, inbred or outbred animals. Thereby, we get a valuable information and shows a preclinical pathway, the development of new drugs. And this particular session I have taken, instead of taking a research on, I am doing at present research on screening of drugs against adverse reactions. I am using bioflavonoids. Recently, I contacted a company also for clinical studies, and it is under process. And if we find out nutrients, the title of this thing is, and if we find certain molecules naturally abundantly available in nature, but the way in which we identify them is the critical one. If we identify definitely against the adverse reaction, definitely it will be a good invitation by the globe, so that the doctor compliance, patient compliance, and the confidence levels also of the doctor patient will increase when we find this type of drug molecules which are acting against the adverse reaction, like cancer. Several drugs are there in certain class of drugs. If they act against or suppress the activity to the tolerable level, naturally that will be a good invitation. Oh, you also think in such a way that why why not we develop a drug molecule against adverse drug reactions? My dear viewers, I am delighted to be with you for the past more than 2.5 hours. And this is a general talk, but I have given the new trends that are to be touched for the research purpose and given certain hints. Those persons who catch them definitely can go for that, because they are all live projects. They are all live projects. And for the degree and master degree and research scholars persons, I advise, you have seen the role of animals in the drug discovery and development and the role of pharmacology. So, keeping in view these observations, I wish you people will carry further research in a more disciplined way to be a good research-oriented personality, to be a good research-oriented personality, to develop, to enrich the science in future. I thank Annamacharya College of Pharmacy and the management and the staff members who are participated in this program. I thank them all. I thank the viewers. I wish you all success in your future endeavor. Back to convener. Any questions? You please.
Thank you, sir. Thank you
Any
for your
questions? Please
thank
ask.
you for your
Thank you. Thank
for
you.
your thank you for valuable information, sir. So we got a very good knowledge from basic pharmacology to advanced technology in experimental pharmacology, sir. Sir, our participants having some questions, sir.
Yeah. Is it possible to cortical spreading depression as anti-migraine model in rats? Say, I am not expertized in this particular model, but that vasodilatation and everything will be there, and hope this model will. The animals are very small. This model may work out, but in my research, I do not have experience on this particular subject on anti-migraine models. But through my experience, I can say that it is possible. It is possible, but you have to struggle a lot. See, while I am developing this model, ischemic heart diseases at 360 beats per minute time, identifying the left descending coronary artery and exactly without damaging the coronary artery, passing a thread beneath it, and finally a double knot technique development, I faced several problems. But we should not leave that particular thing. This mice can be used, but I'm giving my number, the questionnaire. I'm giving Anna Matthews. I'm giving my number. My number, you please note down: 9966729376. See, I will get a model. I will see it through my experience. Also, I will think over. I will give you good answer. Please carry on research. It's a good one. Anti-migraine model that can be carried out, but you have to develop yourself that particular model development. Model development. How I developed in my laboratory. And see, even CCMB people, they came to my laboratory, they learned that particular technique. So nothing is impossible. It is possible. And anyhow, you quoted USA, Brazil, some work is there. I will go through that, what models and how best a preparation can be made and experimental design can be made. I will give you my inputs to you, and you note down my number. I told you, no, 9966729376. Wish you all success, Aparna. It's a good, very good. I appreciate. The problem is very good. You do it because medicine is needed in that particular level. If you give a small scientific valuable information to the scientific community, it is a good invitation. I encourage you, and you be in touch with me so that I will give. Thank you, Amma Subbarani. So today my interest is what? To make you know that are available for the drug discovery laboratory and what type of animals that we are using, and are we using, to question ourselves, are we using perfect scientific animals or not for the purpose of research. And I hope all of you might have got sufficient information and understanding on the basic thing. Thank you. say if you are able to pay more amount, animals, the inbred animals are there. If you take inbred animals, that will become a good model. And I already told you that the diffuse pancreas is there. Now we are conducting experiments using Sprague-Dalley rats or Wistar rats in type 2 diabetes. To the extent they are acceptable, but some more refined animals, as I already shown you, the inbred and outbred. And if your institute or is there, that type of animals can be used. But in initial screening, mice also can be used. In initial screening, if you use these animals, no problem, they can be used. They are also giving good results. Mice also giving good results. Antidiabetic activity, conversions method is there, but that is a screening procedure. But scientifically, in the programmed screening, if you want to extend programmed screening to bring to the maximum level, acceptable level, naturally to the higher end of animals you take, already I narrated, and if you use those animals, your data will be accepted by everyone because the quality animals that you have used. And these animals also can be used, even mice also can be used. In generally, in regular screening at the college level, we are using, and to some extent in research component also agrees on rats, mice. In diabetes, streptozotocin can be used. That. How could we establish a therapeutic window without an animal model? See, these computer operated designs, several things they have come. A guessing therapeutic index, therapeutic window, that is the minimum and maximum concentration levels, can be established, but through animal experimentation only and during the clinical study, if we establish the therapeutic window, because ultimately final dosage settlements and everything will come in clinical pharmacology. It should not be used. I told you, no, thalidomide is a it is possessing teratogenic activity. Thalidomide possessing teratogenic activity. What may be the biggest side effect adverse of vaccine apart from developing the disease itself? Anaphylactic shock. Many times due to the anaphylactic shock, the vaccines, after giving vaccines, people may die. A reaction will be there. Immunological anaphylactic shock could be the reason, biggest side effect with vaccines, and if that particular thing is identified, that cannot be used as a vaccine. What are the different techniques used to target Aurora kinase? Aurora kinase A, different targets, and we have to go and at many places this Aurora kinase A activity, many sites are there, and the research paper, have you go through the Google, you will get information related to that. And you will get the targets, the metabolism, the pathway that are there already described. And if you see that the aurora kinase A at various sites, 3 or 4 sites, the pathways, because it is involved in the proliferation, mitotic proliferation level. Overexpression, when that is there, then it produces cancer. So in the overexpression time, what is the reason? The pathways, several things are given, and you please refer that particular thing. Definitely you will find answer, because cancer research is not my specialization. Drug-induced diabetes and obesity. Certain drugs are there. Sometimes metformin may be induce obesity. Many drugs are there already texted. Many drugs are there. Many drugs are there. When they are taken, they induce obesity. But when it is taken out, again it will come to the normal. Drug-induced obesity is there. Sonia Ankita. Thank you, my dear viewers. I am very happy that you are all followed actual message that I want to give. I have given.
Thank you, sir. It's my immense pleasure to give a vote of thanks on behalf of Annamacharya College of Pharmacy. Sir, you have explained different techniques that are used in experimental pharmacology. So it is very clear. So especially the coronary artery ligation model, it is somewhat difficult. But so your research is published in a reputed journal, sir. So I thank you very much, sir. And you have explained about the different types of receptors and the targets in the treatment of various diseases, sir. Thank you very much. And then your summarizes the pioneers of pharmacology and the role of experimental animals in the drug discovery, sir. And finally, so it clearly explained about the drug discovery process and what the different types of experimental animals, how to identify the different types of experimental animals, and each and every aspect is very useful for the participants, especially it is very useful for the research scholars and academicians. Thank you for your valuable speeches. Thank you very much.
Further, it will be made clear about the involvement in the development of various pathways and disease models. Various vaccines are being prepared by using animal model. MRI is also using animal model. So I have given even mediated mechanism for relaxation.
Yes, sir.
By animal model. Lot of information year-wise also given. I hope all the viewers, if they have those particular slides, that will be very useful for wherever want to give certain reference on animals they use in the drug discovery. Okay, thank you all. Thank you, Dwarkanath.
Sir, thank you, sir. Thank you very much, sir, for to be a part of here. I thank you for all the participants for to grand success of this event, and thanks particularly for Professor Rajeswara, sir, who is having a patience. So in this age also, you are having a lot of patience, sir. For you clearly
Students are my ultimate person. I love
thank
them.
you, sir. And clearly you answered the excellent way to our students. And our faculty members also, they are very happy for this wonderful session, sir. I thank you for once again. And tomorrow morning session will be same 10:20 from Andhra University, Professor. Thank you, sir. So I thank you very much, sir. So now we leave the Yeah, now we leave the session, sir. Thank you, sir.
Thank you very much.
Thank you, sir.
Have a wonderful time.